Can anyone see the slides?
Yes, now we can see your slide. Thank you.
Thank you very much. I would like to say thank you very much for the committee that invited me to present my research with the title Mammalian's Contribution to the Schistosomiasis Japonica Transmission in West Lore Subdistrict, Central Sulawesi Province in Indonesia. Schistosomiasis, previously known as Bilharziasis or snail fever. It is one of neglected tropical diseases that can be caused by the infection of the worm from the genus of Schistosoma that spread in Asian, African, and American continents. More than 200 million people are infected, and annually 10,000 deaths of people caused by the disease. And in Indonesia, the disease is caused by the species of Schistosoma japonicum, and the disease is known as Schistosomiasis japonica. Historically, firstly discovered in Lindu in 1937, and next in 1973 in Napu, and 2008 in Bada. And due to the roadmap of schistosomiasis eradication in Indonesia, Indonesia will be free of schistosomiasis in 2025. This is the worldwide distribution of schistosomiasis. Uh, there are several species that cause infection in humans, and 5 species are cause the infection in humans, and the disease caused by Schistosoma japonicum spread in 3 different countries other than Indonesia are Philippines and China. And this is Indonesia. The disease in Indonesia is only limited into one central province of Central Sulawesi here, only in one province. And the disease is zoonosis, as some reservoirs are cattle, cats, dogs, horses, pigs, and rodents. And it is known that non-human mammals are hosts that can be a source of transmission and also can threaten human health. The infection by this Schistosoma japonicum may show symptoms such as a decrease of appetite, diarrhea, and weight loss. And in animals, animal deaths can occur in severe cases. This is the clinical signs of humans infected by Schistosoma japonicum, namely emaciation, acid test, and hepatosplenomegaly. And this is the sign of infection in animals, cachexia. In China, previously domestic animals known as the main players in the transmission of Schistosomiasis japonica, and buffalo identified in the transmission 80% in an area. And Gray et al. Predicted that the displacement of buffalo from an endemic area can reduce the parasite reproduction rate to below 1, and a comprehensive study in the control of schistosomiasis aimed at inhibiting the transmission of Schistosoma japonicum from cattle, buffalo, and human to intermediate hosts has successfully carried out in China. And this is, I would like to refresh the life cycle of schistosomiasis. It consists of definitive hosts, the mammalians, including humans, and more than 40 species of mammals, and also the intermediate host of Oncomelania snail. Humans and other mammalians are infected when the cell carrier penetrates the skin of the host actively, and it become the juvenile worm, and will become an adult in the liver of the definitive host, and the parasite will move and pair the female and the male worms are pairing and will enter the mesenteric vessel of the bowel, and they will lay eggs, and the eggs will be retained in tissues such as intestinal tissues and other tissues such as uh, lymph node, and can also reach the brain, and some will be excreted through the feces and continue the life cycle. And the mericidium that came out from the ovum will actively penetrate the uncombinaneous snail to uh, complete the life cycle. It is a complex life cycle, and each species of the definitive host has the potential to disease transmission. Until now, there is no commercial vaccine available for the disease. How is the condition of schistosomiasis in Indonesia? Until now, there is limited studies regarding the role of each species of definitive host in the transmission, and currently the role of mammalian animals in the disease transmission is not unknown yet, and infected animals can be source of transmission of Schistosoma japonicum X to the environment and source of reinfections in humans. And in Indonesia, there are 28 villages in two sub-districts, namely Sigi and Poso districts, are endemics, and only in one province, Central Sulawesi Province. And this study will be conducted in 6 villages of West Lore Subdistrict of Poso district. West Lore is well known as Badak Valley, and previously firstly reported as endemic area of schistosomiasis in 2008, and until now there is no case of non-human mammals in West Lore, except one point survey. They found 3 rodents, namely one Ratus novericus and 2 other species infected with Schistosoma japonicum. Thus, this is the map of the endemic areas of schistosomiasis. Namely, this is Lindu and in Sigi district, and also Napu and Bada in Poso district. The aim of the study is to measure the prevalence of schistosomiasis in mammals and the intensity of infection, and also measure the contamination index and also relative contribution index of mammals living in West Lore Subdistrict. And also we study secondary data, prevalence of humans in Lengkaka Schistosomiasis Laboratory with the permission of Poso Health Office. We also do the map drawing. We tag the location of the animal fecal samples and also the location of snail habitats. And we did the collection of fecal samples from animal. Each animal was taken 20 grams of feces of each animal and stored in the cool box. And the animal basis was examined using Danish Bilharziasis Laboratory technique. The technique is the combination between filtration and sedimentation. 5 grams of feces were homogenized with 50 milliliter of saline, and then the solution were filtered using multi-level filter, namely 400 micron, 100 micron, and 45 micron. The retained sediment were then precipitated in dark area, and we centrifugated until we got the uh, volume 2.25 milliliter, and from this we count the schistosoma eggs from 3 different slides. So it's the total for this is if we count 10 eggs, so the egg per gram of feces is 10. And this is the examination of human stool samples using the Kato-Katz from the preparation until the thick smear of the feces. And the measurement is cattle and buffalo's fecal is 25 kg for 10 kg for horses, and 0.5 kg for pigs, and 0.15 kg for dogs. And the total daily fecal output were calculated using the formula, this formula. And also animal contamination index were calculated using the previously published formula, and relative transmission index of each species were calculated from previously reported published formula. And the data were analyzed and presented descriptively. This is the relative transmission index of Schistosoma japonicum. Namely, the total sample of animals are 209, and 1,852 humans stool samples are examined using Kato-Katz. All of the horses, 2 of the horses are positive with the Schistosoma japonicum, and only 0.59% of humans infected by Schistosoma japonicum. This is the prevalence of other species such as cattle 55.55, and pigs 51.16%, and buffalo, and also dogs. So the relative transmission by cattle are the highest, 69.74% compared to others. Pigs 21.95%, and other species only contribute lead in low, and the lowest one is dog 0.31%. And regarding the intensity of infection, humans in average 85.1 eggs per gram of feces. This is relatively still in the low intensity of infection due to WHO, and this is each species of animals, the intensity of infections, and the lowest one is buffalo. And this is the distribution from the 6 villages examined. All of the cattle in West Lore Subdistrict are infected, while in buffaloes only 1 of 3 collected are infected by the parasites. All of the horses come from Tuare and infected by the parasites. And in Kagroa, 80% of the sample pigs are infected, as the highest prevalence in the animal species. And in dogs, and prevalence in humans, the highest in Tomehipi village. And this is I show you the prevalence and intensity of infection and also animal contamination index. It can be shown here that cattle is the highest due to animal contamination index, uh, both individually and also total animals, and dogs are the lowest one. Okay. This is the maps of the green one is the snail focus that is positive, can be found Cercaria. This is the data came from the Sisto government that previously published, and the black one are the location of animals that are positive with Schistosoma japonicum due to this study. The discussion from this, we got that 45.46% of animals collected in this study are infected with Schistosoma japonicum, or with the variety of prevalence from 28.57 until 100%. It suggests that Schistosoma japonicum is zoonotic, that also infect mammals other than humans. Although previous studies, such as Rosmini et al. At 2010 didn't find infection of Schistosoma japonicum in 90 animals examined. This is the total number of each species. And also Anastasia didn't find any Schistosoma japonicum infection in the same area of the study. This can be occurred due to different method of the examination. This study used Danish Bilharziasis Laboratory, while other studies use formalin ether concentration method. And other studies such as Rasmini et al. Didn't find any infection of Schistosoma japonicum from 14 trap and necrosis rats, and there is no infection in non-human mammals until Nurwidayati firstly reported from 3 positive rodents. And this study will add the gap of information regarding the contribution of non-human mammals in the transmission. Okay. The cross-infection between the definitive host can occur depending on the access of each species of mammals to the snail, Oncomelania opensis lindoensis, as the intermediate host, where the infection is transmitted to the definitive host. And the dynamics of and complexity of the transmission is influenced by cultural factors and also local biological factors. And also there is species sensitivity, such as cattle and goats are more susceptible to infection than buffaloes. A human prevalence reported in this study is 0.95% of 1,852 examined. The prevalence is reduced 90% compared to the survey in 2010 and 2008. That firstly, discovered the disease in the area, and the survey coverage is 73.49%. And this study showed that cattle is the main contributor to disease transmission in the village in the sub-district of West Lore. In fact, animals are the vital source of infection and play a role in the spread of the disease. Uh, yeah, that is contamination of Schistosoma japonicum egg to the environment. And animal prevalence reported in this study was lower than reported in Philippines and Coronado, 58.2%, and also in Northern Samar Province, but higher than our previous study in Lindu Subdistrict with 35.8%. This higher prevalence can be caused because no treatment of anti-schistosoma drugs for animals living the endemic areas of West Lore Subdistrict. And our study of the map showed that positive animals were infected during their activities such as grazing, wallowing, drinking, or defecation around the habitat of snail intermediate hosts. And studies on the role of wild rodents in the transmission of schistosomiasis are still needed. This is regarding the intermediate host. Previous studies, a survey of the snail in West Lore conducted by the researchers of National Institute of Health and Research and Development of Donggala in 2008 found 21 poki, and this is in each village. But from the focus found there is no Cercaria was infected. And 2 years later, Rosmini found that 229 intermediate hosts from 4 villages. And from this study, 1% of the snails from Kageroa and 40.3% from Langkeka were found to be infected by Schistosoma japonicum because they found Cercaria from the snails, but no from Tuare and Kageroa. And previous study regarding the snail also reported, Wijaya reported that 18 foci of Oncomelia cupensis lindoensis in West Lore. This is the total area of each village. Thus, this study we concluded that Schistosoma japonicum was found in humans and also in animals, especially domestic animals, cattle, buffaloes, dogs, pigs, and horses in West Lore Subdistrict. And the main contribution of Schistosoma japonicum egg contamination in West Lore is cattle, followed by pigs and buffaloes. And schistosomiasis prevalence in animals is higher than in humans, and we suggest that Praziquantel as anti-schistosoma drugs still need to be given to humans and animals as an effort to control multi-species disease, which aim to prevent contamination of Schistosoma japonicum eggs by the definitive host to the environment. And endemic residents who are at high risk of infection with Schistosoma japonicum must have good knowledge about the danger of the disease. This is some pictures of the endemic area of Schistosoma japonicum. This is the swampy area. Okay. Thank you very much for your attention. If there is any question, I hope I can answer your question. Thank you.
Thank you very much, Dr. Novericko. Now we are at the Q&A session, and there was one question in the chat box. Are there any schistosoma species infected to mammalian animal in Indonesia?
Okay, thank you very much for the question. Are there any Schistosoma species infected to mammalian animal in Indonesia? Yes, there are any Schistosoma species other than Schistosoma japonicum. There are also other species of Schistosoma, namely Schistosoma incognitum and foces. But because my study is to find the contribution of Schistosoma japonicum that also infect humans in Central Sulawesi Province, and to find that if mammalian species other than humans living in the endemic areas also uh, contribute to the transmission. So I hope it answering the question.
Thank you very much. Are there any questions from the floor? Now there is no question, so we thank you very much, Dr. Novericko, for your very informative talk. Thank you so much.